All right. Hello and welcome back to another section of Anatomy and Physiology at Arizona Western College with your professor, Dr. Sean Clavel. And we're going through the sections of chemistry. We talked a little bit about inorganic compounds in the previous section. We're going to talk about organic compounds, particularly those that are important to the human body. We're talking about carbohydrates, lipids, nucleic acids, and proteins. So the things that we consume, and as well as the things that make up us, right? So let me grab my slides and let's get to it. This one. We should first talk a little bit about the way that these organic compounds are either built up or taken apart. Now, particularly these organic compounds, I should give us a little bit of a preface here, have the basic the basic construct of a this idea of a hydrocarbon. A hydrocarbon is a hydrocarbon chain is a chain of carbons, right? You can see here the carbon atoms in a chain here that is then surrounded by hydrogen atoms. And this is kind of the simplest form, as you can see on the top of the slide there, right? The simplest form of an organic molecule. It's called the hydrocarbon. And so there's a large family of these organic molecules that are composed of these hydrogen atoms bonded to a chain of carbon atoms. And sometimes you might see it as like the ball and stick model that looks like this. But ultimately, these organic compounds tend to be what's referred to as polymers. Polymers are like long chains of similar units. So they're built from these smaller building blocks, or what may be referred to as on the screen here, monomers. And so these monomers, these small building blocks of these larger organic molecules, are synthesized, right? This anabolic process that requires energy, they're synthesized by a term that we call dehydration synthesis. And so in dehydration, actually, let me just, all right. In dehydration synthesis, what we have is the one group or one monomer providing a hydrogen ion, one group providing a hydroxide ion, and as a result, water is removed from the equation, and those monomers create a polymer, which is linked by a covalent bond. And so, as a result, many compounds are soluble in water. Many organic compounds are soluble in water, meaning that if you insert water, so you've got the polymer here of a, let's say it's a carbohydrate, right? Like a simple sugar. You could have glucose. Oh, no, come on, let's go. You could have glucose and fructose, right? And so what happens here is that even that polymer, that disaccharide, there's too big to make it past the cell membrane, and so water acts in a way to break the bond, right? Hydroxide ion is inserted to one monomer, the hydrogen ion to another. That is referred to as hydrolysis. Hydrolysis is the splitting by water. So lysis refers to split, to break, to rupture, as we'll see in a number of different sections throughout the chapter here, throughout the semester here. And so hydrolysis is the splitting by water. And so if you place water in a, if you place a solute like sugar into a solution of water, again, as you can see here, you've got a negative portion that'll attract to one part, a positive portion that'll attract to another. It'll break up that bond. It's called hydrolysis. And so, just to kind of backtrack there, right? Dehydration synthesis is the way by which monomers, these small building blocks of organic compounds, are built, and hydrolysis is the way that they're broken down. Keep that in mind. See a star there? Note it. Let's first talk about our first organic molecule, which is the carbohydrate. A carbohydrate, the term carbohydrate itself actually means a hydrated carbon. And so carbohydrates contain carbon, hydrogen, and oxygen, and they do it in this unique ratio. So 1 carbon for every 2 hydrogen for every 1 oxygen. And there can be a number of repeating units of that combination. So carbohydrates are often referred to as saccharides. That's a term that means sugars. And there's going to be 3 types of carbohydrates that we're going to talk about that are important for the body. These monosaccharides, which are actually the monomers, like we've mentioned, right? So those will be seen in our next slide. The disaccharides, which are the 2 sugar units, these made up of 2 monomers. And then the polysaccharides. Polysaccharides are polymers that consist of hundreds or even thousands of monomers. And so in the bottom of our screen here, what we have is, in fact, a ball and stick model of a glucose molecule C6H12. Let me write that up there, right? Because it does follow this 161. So glucose being C6H. Should type it 12 O 6. That's this right there. That's that ball and stick model. So you can see identifying the carbons here. Nice carbon. It's saturated by all of these hydrogens. And occasionally, there's enough, there's a similar amount of oxygen as there is carbon. So important in terms of the human body, the importance of carbohydrates themselves is they're the primary fuel source for the body. So it's highly regulated in the system. They are used in terms of it's stored in the muscle as well as in the liver. It's in the bloodstream, which is regulated to provide enough nutrients, energy to any of the systems that are needed. So the 5 important monosaccharides here, I'm going to do this in these next slides, include the hexose sugars, so the 6 backbone sugars, and we'll take a look at those in the next slide, and the pentose sugars. So monosaccharides generally contain somewhere from 3 to 7 carbon atoms, and they're used to assemble larger carbohydrates such as the disaccharides and the polysaccharides. These first 3 monosaccharides, the hexose sugars, are actually isomers. It means that they have the same chemical formula but a different structural arrangement. And so the hexose sugars, these C6H12O6, glucose, fructose, and galactose, are really kind of like our dietary sugars, right? Like our dietary sugars, the sugars that we use for energy and that we eat. The pentose sugars are the are constructs, or how do I put this? The pentose sugars are the sugars that we see in the nucleic acids. They're made in the nucleic acids here. So what we're talking about is the makeup of the monomere of DNA or RNA is a nucleotide, and so the pentose sugar, deoxyribose nucleic acid. The pentose sugar here is the portion of DNA. Then there's a polar phosphate group and a nitrogenous base. So this is what the makeup of DNA is, the single nucleotide is monomere of DNA. And so we see deoxyribose in DNA. And then we see the ribose sugar in RNA. So those two pentose sugars are important in the nucleic acids or the nucleotide structure. Disaccharides, or these two sugar units, are simple short chain carbohydrates. There are three common disaccharides that we should be familiar with. So the three common disaccharides include sucrose. Now, sucrose is a combination of the monomers glucose and fructose. This is a transport form of sugar. It's used by plants and harvested by humans for the use in food. Lactose. Lactose is a combination of glucose and galactose. It's present in milk. And finally, a maltose. Maltose is the two glucose units that's present in germinating seeds. So again, the three important disaccharides being lactose, sucrose, and maltose. And then the three important polysaccharides. So the polysaccharides can be these long or even straight branches of hundreds, maybe even thousands of these sugar monomers. And these polysaccharides are also called complex carbohydrates. So starch is a plant storage form of energy. It's arranged as unbranched coiled chains. As you can see there, the amylose, just a long chain that might be coiled. Or it could be in this amylopectin form, which is, again, this branched form. Glycogen is the animal storage of glucose, or of carbohydrates. So these are highly branched chains that are used by animals to store energy in the muscle and the liver. And then finally, the cellulose, which is a long, fibrous form of carbohydrates, which is a polysaccharide, which creates the cell walls and is indigestible by humans. And so there we review our glucose metabolism. In the next section here, I'll talk about the lipids, followed by proteins and nucleic acids. But we'll take a break here. In the next sections, we'll talk about some of the structures of the other organic compounds, which include, again, the lipids, nucleic acids, and proteins. But again, I want to just share this slide with you here as we can see like the intake. Oh, where'd it go? Just click too fast. The intake of glucose metabolism is starches, glycogen, and disaccharides, as well as some of our monosaccharides, glucose, fructose, and galactose. So we intake all of these. We store it in the form of glycogen, and then it's distributed and utilized in the form of glucose. Glucose is the utilization form. But like I said, in our next section, we'll talk about lipids. We'll take a break now. Thank you, and take care.